Oh, recording started, okay. With a model that we call GmGM, that is Gaussian Multi-Graphical Models. And this is part of the PhD project of my student, Andrew Bailey, that is co-supervised also with David Ristek, Professor David Ristek. Okay, so this is our structured talk. I will give a little bit of a background on Gaussian graphical models, and then I will describe our approach that was recently published in iStats 2024. Okay, so background on Gaussian graphical models. What is a Gaussian graphical model? It is just a multivariate Gaussian distribution when we are in multidimensional space. So it's completely specified by its parameters, that is the mean vector and the covariance matrix. But what we are actually interested in is the precision matrix that is defined as the inverse of this one. Here, in the context of omics, you can think that the number of features, for example, is the genes or the number of cells. So let's focus, for example, on the number of genes, for example. In this picture, has 2 dimensions. So what is interesting here? So what makes that a graphical model is not clear yet, because we need a graph. We need to understand where is the graph. So in our context, the variables are the vertices of our graph, and the edges would be defined by the non-zero elements of the precision matrix, the support of the precision matrix. So what's interesting about the precision matrix is that it only describes the direct interactions between variables, not the indirect interactions. So, for example, the covariance will tell me that x1 and x5 are related, so there's a covariance between them, but actually those nodes are conditionally independent. So if we condition on x2, those are independent. And this is the Markov property. And this is what happens here in this matrix, that we see there is no, so there's a 0 in the element 1, 5, for example. Okay, and that's the concept that we're going to stick to. So this is another example, and if you heard about partial correlation that was introduced by Dempsey, it's a similar concept that they also explained starting from the precision matrix. So it's a more standardized version, let's say, of that. And in this example, in a hypothetical example, derived with the GmGM on psychological variables. So the fatigue is causing insomnia, and the fatigue is causing concentration problems, but then the correlation will measure a correlation between concentration and insomnia, but actually this isn't what we're after. We want to make sure that we're only modeling and estimating the direct interaction. So in this case, this 1, 3 element or the partial correlation has to be 0. Okay, because there's no direct link. And the conditional independence implies then sparsity. If something that wasn't 0 with the correlation would be 0 with the partial correlation or with the precision matrix. That's the idea that we want to exploit. So the support of the precision matrix will express the dependency graph. And a classic example is through a random walk. So if you start from a random walk and you have a IID set of variables that are known as 0, 1, for example, you define your variables and your future vertex in your graph as with this rule. So each one depends on the previous one plus this ZI. What we have here is that the covariance is a full matrix, and this will correspond to a full fully geometric graph. But actually, if we estimate the precision matrix is an easy computation. What we get is a three-diagonal matrix. So this is much more sparse than the covariance, and actually will correspond to a very much simpler graph that is just a straight line. So just to build the idea of what we are trying to do and the kind of problems that we want to focus on. These are toys examples, of course. So we want to study Gaussian graphical models where the precision matrix is sparse. Okay, and this means that we are expecting to have more condition-independent variables. And the advantage of this is that when we have fewer elements to estimate, we also need fewer data. Okay. Now, the sparsity assumption implies that the maximum degree of our graph is smaller than the number of features, because not all the variables are expected to be connected to the others. So because the variables are nodes, so the degree cannot be P, so much smaller than P actually. And this is reasonable in many contexts, and in particular in gene interaction networks. So this is an example of a gene interaction network that is being built in this work, cancer genetic network inference using Gaussian graphical models. And here there were 15 cancer networks with 10 normal networks from the cancer genome atlas. And so these were the expression data that were used. Then some subset of genes were considered extracted from the KEGG cancer factors database, and then this network was compared, and those are the links that we found. So as you can see, this is not a fully connected graph. So this corresponds to the assumption that we were looking for, that there is sparsity in the precision matrix. So this is the kind of problem, but we are going on a much larger scale. Now, so we like sparsity. However, it's convenient anyway, because there's no procedure that does well in dense problems. So, and this is the vector sparsity principle from this line. So anyway, it's a good thing to assume sparsity, because we let us also work better. Now, how do we ensure sparsity in this context of Gaussian graphical models? So the first to introduce this sparsity. So sparsity was imposed by Friedman, Haste, Tipchiran in 2008 with the graphical lasso, using an L1 penalty on the maximum likelihood estimation of the precision matrix. So that's this term here, and that's how the sparsity was enforced. So this is great, this worked, but of course there are some limitations. Because when we have a complex problem, like for example, the one we are looking at in biomedical applications, so you can imagine gene expression and cell expression. In this case, we are assuming the independence between features and samples. So we are only estimating one graph in one, let's say, dimension, not in the other one. Because, of course, we are assuming the independence in this dimension, and then we estimate the graph along the features, for example, the gene interactions. But if we then go on the other dimension, the samples, then we go against the previous assumption that there was a conditional independence. So in practice, we can't assume that then the features are independent. So we cannot proceed in one direction and then in the other, because we made an assumption. So we can't contradict this assumption. So we find a graph between features, for example, and that's a model choice, right? So we want to remove this independence assumption. how do we proceed with graph estimation without independent assumption? So again, it's a model choice issue. We don't want to make this choice. We just want to have a more general framework where we can, at the same time, estimate a graph along the features and along the samples, for example. And this general framework should be able to estimate a sparse graph that is interrelating features and data points or samples. And there was the first attempt that, as far as I know, was from Kalaitzis et al. In 2013, where the kind of problem that we can summarize is, for example, they managed to address is a rotating image. So you can imagine that this is always the same dot that is rotating. Or you can, so you have one dimension is the frames and one dimension is the pixels of the image and images actually. And or you can have genes and cells, as I was saying before. So we want to extract the conditional independence between gene and cells, for example, in this case. And how they did, they used a specific instrument that is a chronograph sum that preserves the matrix structure. So in practice, there's a precision matrix that is estimated along the rows, a precision matrix that is estimated along the columns, or vice versa, so the 2 dimensions. And the overall precision matrix is defined as a chronograph sum between the two. Okay, so this translates into the Cartesian product of the graphs adjacency matrices. This is the model. So it should be a product using the this sum. So this handles the problem, but the way this was implemented actually had issues, because even if the space complexity should be n squared plus p squared, because that's what we have to memorize, not consider any sparsity, of course. Actually, when this was implemented, it was implemented to store the overall thing. So it's n squared plus times p squared. So this also implied a very slow computational time. But the main limitation was the space complexity in this algorithm. And these could only handle tens of samples. So solve the problem is a very good idea, but it's impractical, not for the kind of problems that we want to address. So later on, with another previous PhD student, Sirja Lee, we exploited the eigenvalue decomposition of that Cartesian product, and we managed to have a more efficient version of their algorithm. And this reduced actually the memory requirement to what we really wanted. And this is an example of application on a mouse embryonic stem cell data set with 182 mouse embryonic stem cells and a subset of genes involved in the mitosis. So on synthetic data, we had very good accuracy, and also in real data, everything that we had here made sense, because this was checked with the biologists. But we didn't solve completely the problem, because still we stepped up, let's say, because we were able to work on hundreds, let's say, of points, but not on thousands. So we improved the memory requirement, we have improved the runtime, but this is not good enough for the dimension of the problems that we want to solve. So, and we call this one scalable big class. Now, there are other approaches. One is called Terra Lasso, for example, one is called A-class, that have a better space and time complexity. So this is their runtime, for example, and can run on low thousands in reasonable amount of time, that is here, okay, in minutes. Now, limitations, this doesn't scale to millions of features. Those are iterative algorithms. That means that because every iteration has got a negative composition that has a n to the power of 3 runtime, those are slow. And the memory usage, it's okay. It's what it has to be, because it's n squared that actually is needed to memorize the data. So can we do better than this in terms of runtime? And the other question is, do we need to scale to millions of samples? So we want to write models that can handle millions of samples, but actually is this necessary? And if we look at the single cell expression atlas, you can see experiments that are not even very recent, that have like 500,000 number of cells. So this is about 500,000, this is about 700,000. If we go now to the human cell atlas, and we find here an estimate cell count of 4 millions to billions. So current approach to analyze this data set, because of this space and runtime constraint, is to split ahead of time, to have some filtering on the data, because there's some biological information, so we cannot actually do it, blindly analyze the whole thing. So this is an idea. So the answer is that we do need to do something about it. Also, this is only going to get better in terms of data availability and worse for the modeling side, because if we look at this plot from Krashenko, 2021, so now this is even old, but this is a very nice plot where we have in log 10 scale the number of transistors for a computing infrastructure, and the log 10 of cell counts. And here we got the a collection of main experiments, okay, with the red line. And here we have the increase, let's say, in the CPU transistors counts. So as you see, there's a certain increase, an irrelevant increase in capacity, in computation capacity, and actually there's an even more peaked increase in the number of cells measured by these landmark data sets with the different technologies that in the years have been developed. And actually, this black line is feeding what is called like the Moore's law, that was saying that this number of actually the transistor counts should double every couple of years, but actually this is a log 10 scale, so it's doing much more than that. So in practice, we have more and more and more data available, and it's only going to grow. And we need to be able to handle that. So do we need to scale to millions of samples? So the answer is yes. I hope I convinced you with those reasonings. So that's why we try to have another step up in the computational time and memory. With this approach, GmGM, they're talking about develop during this PhD of Lady Andrew. Lady is the name, by the way, I'm used to saying. So, and we have a repository for it. So with this, we have even a much general framework where we can work with multi-omics. That means you can think of two, just for an easy example, metagenomics and metabolomics. So you can think of this example here. This is Lifeline's deep data set where they were gut bacteria measured in people, just the same people, and we have measurements of species collected in the streets of these people, and then the metabolites collected from the same from the same samples. So the idea is that those two omics, okay, or modalities, we want to say so, have got a shared axis because the people are the same people in both of them. So it is reasonable to expect that if we proceed towards a graph estimation, this is the same graph. So we don't want to separate the problem like looking at this data set and this other data set and then compare what we got, because that is what is often done. So we estimate networks and then we have a kind of consensus between all the different data sets or the instances of different data sets. But no, we want an enormous, gigantic precision matrix that will embed all the little precision matrix along all the axes of all the modalities. So we're not going to memorize it all, but we are just keeping that in mind when we model and we estimate everything at the same time. Okay. Okay, so that's few details, very, very high level details about the model. So we work with tensors, sharing an axis that will be drawn from a Kronecker sum of normal distribution. Okay. So the idea is that when we parameterize all of these, the shared axis will have in this sum, will have the same precision matrix. Okay. Okay. So we go down here. This thing goes somewhere else. You go down here, you see we have the same precision matrix here and here. So when we model this data, we keep that in mind and we estimate everything at the same time. All the entries of those precision matrices, they will be our graphs. The support of those will be our graphs. So what is the improvement? So in previous works, this L1 penalty was included to enforce sparsity. And those are all iterative algorithms. They use an eigen decomposition every iteration. That makes them slow, as we were saying. But if we remove the regularization, we'll only need one eigen decomposition, and then we can proceed. So instead of regularization in practice, we propose to use thresholding. Now, what do we gain and what do we lose when we do that? So for sure, what we gain is that we are much faster because those are the literature ones that we were mentioning before, parallax and a glass. So this was the graph that we did before, and this is where we are. So we can actually run on a real data set of 10,000 cells under 1 hour, while we cannot even attempt without this will never happen with this other approach. Okay. And I will show you the accuracy now later on. So, but is our approach good enough? So we do reach the space complexity. There is a memory limit of n squared, and we can't do better than that because still we need to memorize the data. We can't get rid of them. And this is indeed optimal for this problem. Okay. But we are working on additional assumption. And this is a work in progress, so this is under review. Now, these additional assumptions are the following. So the input data sets that is an issue because it's enormous. And the precision matrices could be approximated by a low rank version. And then we can also ensure a fixed upper bound for the sparsity of the graph that we want to estimate. So when the dimensions grow, this will not grow with the dimension. And this is an example. If we use 50 principal components with this equivalence of retaining 50 eigenvalues in our decomposition, this is where we end up in terms of runtime. So we have had a massive decrease in runtime. And with this low rank approximation, we also reduce the memory required. And now you will all be asking, so what about the accuracy? What are you losing? Because you can be fast, but if your results do not make any sense, so what's the point? And indeed, there is a bit of a trade-off. So on small graphs, so here on simulated data, okay, so we have Basil-Albert graphs with 250 nodes. So this is the original unmodified GmGM, the one of last year in ISAT that was described in the beginning. Those are the precision recall curves. And we do well together with the TeraLasso, just reporting TeraLasso here, okay, because it's a tensor approach. So we are up here with TeraLasso, we're doing very well. But when we start introducing these approximations, we don't do very well. We do worse, still not terribly worse, but much worse on small data sets. So the suggestion is this is really not necessary, to do. You do not need any approximation. You do not need to use this fast version of our algorithm when we have a small data set. Okay, because you can handle this in a very reasonable time and memory space with the classic approaches, or actually with our approach. The situation change when we have a much larger graph, and here we can't show TeraLasso because TeraLasso it couldn't run because it would take a very, very long time to run. And those are the results that we get. Okay, so still the non-modified, the original GmGM does better, but this isn't doing too bad, this the low rank version, let's say, with the low rank assumption. This isn't doing too bad. And same thing, this isn't terrible. Of course, the performances are decreasing, but also it is a bit of a struggle to generate synthetic data that respect all these assumptions with the low rank. So we are using a low rank assumption that the generated data are not respecting. So the take-home message is we can do well on big data sets, very large. And you can imagine, you can expect a larger than this is getting better and better, but it's really not necessary, and actually it's bad to use these fast versions, this low rank assumption on smaller data sets. Okay. And then there's another issue that I'd like to discuss is like when we want then to test what happens on real data, the problem is that we do not have a gold standard. On large data sets, we do not have a gold standard. So there's no ground truth that is not, everything is interpretable. So we need to make sure that we are actually asking the right questions to the biological sample we are analyzing. And so it's very hard to tell if you did well or badly in your estimation. Anyway, well, this is an attempt of interpreting the results. We estimated, for example, the networks of genes, and then we looked at the GO-terms, and this is a validation on the clusters that we found on this. And it seemed to make sense, but who knows? There's a little bit of agreement between Terra Lasso and GmGM on a subset of these, because again, Terra Lasso has more limitation in terms of runtime. Okay. But again, so there's no, we didn't identify a good gold standard, but of course, if you have any suggestion, we welcome any suggestion you might have. So just to wrap it up, GmGM versus prior work. Now, prior work will need the 440 giga to memorize data, assuming double precision. Ours will need 2 gigabytes. And on these dimensions, prior work will take 2,344 days to run, projected on this runtime of n to the power of 3. And we will take on these dimensions, on this kind of size of input, 20 minutes. Okay. Now, do we need, why is it here? Sorry. Sorry, that wasn't supposed to be there, but anyway. Million cells data sets. Well, I mean, I guess I put it there just to reconnect to the fact that actually we have to keep in mind that we have million of cells data sets, okay? And indeed, there's another problem there. So we only used Bailey's computer to run the experiment that has got a physical RAM. I mean, the RAM is 8 gigabytes, so we can't do better than that. So those are projected numbers, okay? On millions cell data set, prior work will need 16 terabytes for memory. We still need 20 gigabytes. This can be addressed. I mean, it's not the end of the world. So we will need a very powerful machine that we don't have at the moment, or we will need a parallel implementation that we didn't do, but it's very achievable. And this will be the runtime. Okay. And just to conclude, so the next steps, what we are actually looking for is a large gold standard data set. So we would like to have some more certainty when we validate our model on real data. So at the moment we are struggling to have this gold standard. So we keep trying, but we're not very happy. And then we will extend this to directed networks, because this was all for undirected ones. And we're working currently on some modeling advancements of just little changes in observation and the model. And that's what we're doing. So I guess this is all I wanted to tell you. Thank you for listening.
Very much.
Yeah, sure.
I'm sorry. I thought so. I heard someone talking. Okay, so thank you very much for the talk. That was really interesting. And now we can open the discussion for questions and discussion. Any questions? I don't know if I can. I do see a question here for someone that I can't see the name because I'm sorry, this is the webinar first time. So they're asking, what are the precision and recall are here for? What are we predicting?
Yeah, so what we are predicting in that precision reports are the correctly identified edges. Because we got simulated data, we know the truth. Huh? So that's what we did. So yeah, the correctly identified edges in the simulated data. So because when you simulate the data, so it's not, I have to say, the simulation of the data was you have to think, how do I simulate the data? So you have to simulate data with a certain Kronecker sum structure in mind, and so you know which the precision matrix should be, so which the network should be for your data, and then you do your prediction. So you estimate the precision matrix, and then you evaluate the number of successes, let's say, that you get when you identify the right edges. Yeah. Is a hand up?
Yeah.
I have a question. Yeah, I'm a panelist, so I can talk. So I can't post messages. So I just wondered, so on the analysis of the real data, I think the next slide, yeah, there wasn't much didn't look like there was much overlap between the two, between your algorithm and Terra Lasso. Is that generally the case when you look at these things?
So the thing is that it's there's a lot of there are a lot of choices that we have to make when running these algorithms. So we can kind of adjust the thresholds of our one so that we catch similar. So the biological processes were not exactly the same one that we identified, but they all. So what I meant by similarities is that this particular data set is related to the immune response, let's say, measure, and those terms made all sense for this GmGM or for Terra Lasso. They were related to immuno responses. So they all made sense. So they made sense. So those are the ones that made sense, let's say, in our answer and in their answer. So maybe the two algorithms identify different parts of what's the truth. So we don't know. That's when so we are more confident in comparing algorithms on synthetic data. When we go down the route of the real data, it's difficult to interpret the results, because bear in mind that what we're doing here, we're not comparing element-wise the networks. We are extracting clusters and trying to make sense of the cluster. So maybe we could have done something different. Maybe we could have looked at the enrichment. So that's things that we are discussing at the moment. So we could have in mind a list of genes and then compute the enrichment of those genes in the two networks. The one estimated with the GmGM and the one estimated with the Lasso, for example, rather than going in validation in gene ontology terms. But yeah.
Okay, thanks.
Any other question? I do have a question. Very interesting talk, and very I would like to see where this is going. I do have a question about the principal components. I imagine for now it's an you choose arbitrarily 50 or 250 and whatnot. Would it be possible? I have no idea. No, maybe a naive question. But would it be possible to start with a very low number of principal components and just have the algorithm be like, okay, just keep on increasing the principal components until, or either the increase in explained variability is this critical value, or if the time surpasses this, then cut it over, and then you can start like thinking.
It's a very good point. It isn't totally arbitrary. So mainly we did use what was more convenient in terms of, so sometimes it is like, this data set is enormous, the most I can do is this, and I cut there. you know, because you can imagine that you want to keep the highest one, but still make it run in a reasonable amount of time, right? So so you want to trade off. So you do not, if you can avoid the low rank approximation, that's ideal. But if you have to use it, it's you could project on how much you're willing to wait. I guess. But in terms of accuracy, you could have a guess based on the simulation. Yes, I mean it makes perfect sense what you're saying. Indeed, it is arbitrary. So
Great. Thank you very much.
I think
Any other question? Yes, we can. I'm gonna talk to Dr. Luisa Cutillo. Sorry, I have a question. Someone asking if we're gonna share the slides of the recording. So
yeah.
I did record it, but I'm gonna ask you later if it's okay for me to share or not. If it's okay, it's gonna be on the website, in the industry group website.
Yes, sure.
Thanks. Well, if there's any no further questions, I want to personally thank Dr. Luisa Cutillo to make the time and having this an very interesting talk.
Pleasure. Thank you for inviting me.
And I hope we figure out this webinar series technicality problems for the next time, next month. Please stay connected. Andrew, yeah, please. There's someone with a raised hand. Oh.
You just can make him panelist and then he can talk.
Yeah, and I allowed him to talk. Andrew, if you, I think you can. You're muted. Nope. Oh, wrong button. He pressed the wrong button. No worries. No, please. Thank you. Thank you, Andrew. All right. See you, everyone, and not in December. I mean, we're in December and in January for the last Wednesday of that month for the next talk.
Okay.
Thank
Thanks a
you.
lot. See you. Bye-bye.
Thank you. See you.
Stop recording.